Hi, my name is Mackenzie, and today we're going to talk about Watson and Crick DNA structure. First, let's talk about the basics. We know that we have 5 nitrogenous bases, which are information carriers of genetic bases. We have 2 categories, purines, which are adenine and guanine, and pyrimidines, which are cytosine and thymine. Deoxyribose sugar molecules are second, and they are held together by a phosphodiester bonds. Sometimes we call these phosphate backbones. Let's talk about this in a little more detail. So adenine pairs with thymine, which are held together by 2 hydrogen bonds. Guanine pairs with cytosine, which are held together by 3 hydrogen bonds. Hydrogen bonds are weak attractions between positive and negative charged molecules, otherwise known as purines and pyrimidines. Our overall DNA structure. So as we can see, the black strands represent our anti-parallel strands, meaning that they never touch. Our ribose carbons. Our first carbon is our 1 carbon, which attaches to the base. Our second carbon distinguishes DNA from RNA. So DNA will have a hydrogen and RNA will have an OH. Our third carbon bonds to the next 5 phosphate, and then our 5 carbon contains a phosphate. Extra information. We read and write DNA from the 5N to the 3N. DNA-based stacking is used to maximize hydrophobic interactions, and hydrophobic interactions are the main force that stabilizes the double helix form. Major and minor grooves. Major grooves are 3.4 nanometers and means that the backbone are far apart, while the minor is 0.34 nanometers and the backbone is close together. Watson and Crick's DNA is B DNA, and it is always a right-handed spiral. Denaturing. When DNA molecules are placed in a higher concentration, such as saline citrate or SSC buffer, denaturing of the DNA will occur, meaning the hydrogen bonds and hydrophobic interactions will break or stop, therefore the 2 strands will separate. You can demonstrate the denaturing of DNA using a DNA melting curve. Tm is the midpoint of the thermal denature. A melting curve shows the Tm and the possible configurations throughout your denaturing process. Thank you for watching.